Hello, everyone. I'm Aishwarya Nambiar. I'm a graduate student in Tim Miller's lab. And so far, we've heard a lot of different aspects and angles and approaches to this ALS research question. And how do I go next? Oops. Okay. So for the 50th time today, showing you that as Tim spoke about DNA, your DNA has genes, which is basically an instruction manual that has information and encodes all the information for all the proteins in your body. What I wanted to highlight here is that your cell uses this RNA, which is your messenger here, that carries this information and takes it to machinery to make all these specific different proteins. Sometimes, however, we have errors in your DNA that can call gene mutations that can cause you to have either too much of a protein, too much of normal protein is harmful, too much of the wrong version of the protein is also harmful, but also too little of a protein can be an issue too. So for my talk, I'm focusing on this portion, which is the insufficient amount of protein. And an example of that is a gene which codes for TBK1 has this loss of function mutation where the broad concept is you have only half the amount of protein than what is required for a healthy functionality. So imagine protein insufficiency if half is a problem, half amount of protein is the problem, we would assume that restoring or boosting protein to recover that protein might be therapeutic in such scenarios. So we turn to our very trusted antisense oligonucleotides that are being used in the clinic in various different capacities to treat many different diseases. And we've definitely heard about tofersen today, and that is very exciting prognosis. So keeping that in mind, we were how do we use these ASOs to try and increase protein expression? So I've thrown in a lot of descriptive words about ASOs, but the main takeaway from this slide that I want you to look at is that ASOs can target your messenger, that is the RNA. So by doing so, it can either get rid of bad protein, which is what is done with tofersen, or choose it to ask to make the right form of protein. But what hasn't been, until recently, been used for or applied to ASOs is for loss of function, how do we use ASOs to boost protein expression? So that is basically the crux of my project where, imagine in your mRNA, you have certain sequences that can bind to something that you saw in Dr. Galloway's talk about microRNA binding to your RNA. So when a repressive element binds to your RNA, you can get rid of it destroys the RNA, you have less amount of protein, so on and so forth. They have valuable use in day-to-day cell function. However, in conditions where you have little protein, we predicted, what if we make an ASO that can mask this region such that the repressive element is not able to bind to its sequence? That would mean we could stabilize the mRNA, stop it from being degraded, and maybe have more protein. And this approach allows us to make a new, let's say, a class of ASOs that can boost protein expression that could be broadly applied to treat diseases that can caused by low loss of protein. Thank you.